In the second part of this lecture video, we'll be looking at blood pressure regulation through the vascular system. And so we'll begin by talking about a term, vocabulary word for you, called vessel compliance. The compliance of a vessel is an indication of how well that vessel can stretch or extend out. And so in general, veins, your veins tend to be more compliant than arteries, mainly because they have a little thinner muscular wall. And so compliance in a vessel, you kind of have a little flow chart here, I guess. If compliance in a vessel decreases, if the ability of the vessel to stretch out or to extend out or distend decreases, generally that means that blood pressure will increase. And this table, or this figure, figure 18.5, gives you kind of an idea of various vessels that we've talked about and how blood pressure changes or is kind of changes in each of these areas. And so look at, for example, the aorta. This is, of course, is your largest of your vessels, and they're kind of listing these out in the order from highest pressure to lowest pressure. And so the aorta happens to be the largest of your vessels. Of course, this is an artery, and you can see here that it has a highest amount of pressure. The line here represents the mean arterial pressure. And you can also see the changes in systolic and diastolic pressures in each of the areas. And so as you start moving out the elastic arteries, the pressure there is pretty close to what it is in the aorta, maybe slightly lower. And then the muscular arteries go down a little bit from that. Then remember, the arterioles are smaller in size, and so you see a big decrease in blood pressure once the blood reaches the arterioles. Then capillaries still have a little bit of blood pressure there, but it continues to decline very rapidly. And of course, by the time you get to arterioles, there's really no fluctuation in the systolic versus diastolic pressures. And then when you get to the smaller of the veins, the venules and the veins, then blood pressure really drops off to get to the vena cava. Now, regulation of blood pressure through the vascular system can be short-term, and so these are some examples of how your body is trying to regulate blood pressure through the vessels. And so part of that deals with your nervous system, your sympathetic nervous system. And so if you remember your sympathetic nervous system from Bio 168, it can stimulate and cause the release of some hormones that we've already mentioned in chapter 16, norepinephrine and epinephrine. Of course, these come from the adrenal gland, from that medulla section. And so when these get released, they target certain cells, and they can cause, for example, your cardiac cells, your cardiac muscle cells, to increase the heart rate. And those are some of the factors that some of these hormones can do. Increases the heart rate, that speeds up, and the contractible force, or the contractility of the heart, basically how hard the heart is actually pumping, that increases. The force of contraction increases, so we're talking about an increase in blood pressure. Your sympathetic nervous system generally tends to increase your heart rate and blood pressure. It's your fight or flight scenario. By increasing heart rate and increasing blood pressure, you increase cardiac output, just like we've talked about in chapter 17 on the heart. But it can also have other areas as well. Vasoconstriction, causing blood vessels to constrict, that increases resistance. That will increase blood pressure. So as the vessels get smaller, and most of your vessels, especially your arterioles, your smaller vessels, vasoconstriction, that'll increase resistance, and that increases the pressure there. These hormones, norepinephrine and epinephrine, usually cause some vasodilation of some of your vessels, such as the vessels going toward your skeletal muscle, the vessels going to the heart, and that allows for more blood flow. They dilate, so they expand. The diameter increases, and so that allows for flow to increase to those areas such as your skeletal muscle and heart. And again, this is fight or flight, because this provides more blood flow, more oxygen, more gas exchange to those critical areas such as skeletal muscle and your heart. The parasympathetic stimulation, if you remember talking a little bit about parasympathetic nervous system in 168, parasympathetic stimulation usually slows things down, and that's what it's going to do here. The vagus nerve has an inhibitory effect on the heart. It decreases the heart rate, and remember that's because of the acetylcholine gets secreted. It actually hyperpolarizes the heart. That means it takes a little bit longer for that action, for the next action potential to be generated. So parasympathetic stimulation slows down the heart rate. You also have, in the short-term maintenance of blood pressure, something called a baroreceptor reflex. Baro is a prefix that refers to pressure, and so this is a reflex action that's trying to, again, control your blood pressure. And so if you direct your attention to the figure 18.8, I've got just part A shown here. It's trying to balance blood pressure homeostasis. And if you start here with a stimulus, you've got some kind of stimulus that causes blood pressure to go up. It goes up beyond its normal range, and that gets detected. Remember that you're going to have a receptor that detects different types of physiological characteristics. In this case, we're talking about blood pressure. And so in this case, the baroreceptor usually is located in some of the vessels around the heart, maybe the carotid arteries, even in the aorta, you have baroreceptors there. These are specialized cells that can detect pressure. And so they can detect that there's an increase in blood pressure. That sends information through the nervous system to the control center, in this case, to the brainstem region, the medulla section of the brainstem. That causes, that's the processing center of the control center of this particular physiological characteristic. That sends information back to the heart, the effector muscle in this case, through the vagus nerve, and that allows for the heart rate to try to slow down, and hopefully that should get your blood pressure back into its normal homeostasis range. All right, so that's kind of the pathway here. Blood pressure goes up, and it sends that signal down, eventually makes its way back to the heart to try to lower the blood pressure. You can look at the opposite scenario in the baroreceptor reflex. In this case, your stimulus is that your blood pressure goes a little bit too low. It's starting to drop a little bit below the homeostasis normal range. Remember, blood pressure is, maybe somewhere around 120/80. So if it drops below that number, it should be detected by, again, the baroreceptors. That sends the information again to the medulla section. But in this case, it's going to block what the vagus nerve is doing, and it's actually going to stimulate the sympathetic nervous system. And that's going to try to increase the heart rate, try to increase the force of contraction, and that will hopefully increase cardiac output and increase blood pressure. Now, other chemicals in the body have an effect on your blood pressure and what's going on in your vascular system. Oxygen, pH, you have chemoreceptors, chemical receptors, just like you have blood pressure receptors, the baroreceptors. You have chemical receptors that are trying to monitor various chemicals in the body. I've listed two of these for you, oxygen and pH. And these are, again, located closely associated with the heart in the aorta and the carotid arteries. And so oxygen, in general, if your oxygen levels start to go down, that generally causes your heart rate to speed up, and it causes vasoconstriction to try to increase your blood pressure. If you have lower levels of oxygen, you need to try to send that blood out a little faster to try to get the oxygen that you do have out to circulate throughout the body. We don't always think about pH as a chemical. pH is a measure of a chemical, though. It's a measure of hydrogen ions. And so whenever your pH begins to change a little bit, if your pH increases, and so that means your pH is actually becoming more basic, you're becoming more alkaline, then your body tries to accommodate that, and it tries again to speed up the heart rate to try to get more oxygen to flow out to the tissues. And then lastly, we have hormones. Hormones help to regulate blood pressure as well. We've already talked about a few of these because epinephrine and norepinephrine both directly impact cardiac output by speeding up the heart rate. That affects cardiac output, but also thyroid hormone. Your T3 and your T4 metabolic type hormones, those also help to increase cardiac output by increasing metabolism. Hormones that help to control resistance, well, that can be your epinephrine and norepinephrine. They both balance out vasoconstriction, and that kind of helps to control blood pressure as well. Angiotensin 2, this is a hormone that we've talked about a little bit. We'll see this hormone again when we get to the urinary system, and fluid balance is very important in fluid balance. It also works here as a vasoconstrictor, and so it gets secreted to try to increase blood pressure. And then ANP, atrial natriuretic peptide, we mentioned a few things about that. We'll see this again as well when we get to the urinary system as well, because it does help to maintain fluid balance and help to control blood pressure. It acts as a vasodilator, and so that helps to open up some vessels and will help to decrease or lower blood pressure. Section 4 really looks at capillaries. We haven't really talked much about capillaries and something called tissue perfusion, and that's really kind of diffusion of blood flow out into the tissues. It's getting that blood out into the tissues through the capillary network that's there. And so you kind of see in the picture here an arterial kind of going through, and so this is carrying blood away from the heart, and you see the connections that it has with very small vessels called the capillaries. You have a capillary bed or capillary network, and then you have a venule, a small vein on the other end. All of the gas exchange and nutrient exchange would take place in the capillary bed, and then once blood has kind of gone through the exchange process, it enters back into the venule on the way back to the heart. Now, for tissue perfusion to take place, you have to have diffusion, and so capillaries have the ability to go through diffusion of different molecules. And so depending on what kind of molecule you're talking about going in and out of the bloodstream will depend on kind of how it passes through the capillary. And so there, if you have a lipid-soluble molecule, just like when we talked about lipid-soluble hormones, you have a lipid-soluble molecule that you're trying to get into the tissues or get in or out of a capillary, it can usually pass through the plasma membrane. Even smaller water-soluble molecules can go through the plasma membrane. But larger molecules, especially larger water-soluble molecules, they have to have some type of opening or passageway to get through the capillary to diffuse in or out. Those gaps are called fenestrae. They're like little windows inside of the cells or in the cell membrane that allow for molecules to pass through. And in this figure 18.11, it shows you some examples of how molecules would pass in and out of a capillary. So that's called capillary exchange. And so you've got a couple examples. So for one example, you have little gaps here between the cell membranes of two adjacent cells. So there's sometimes a little gap there that allows some molecules to kind of pass in and out. The little ovals or circles, these are the fenestrae or the little windows that are here inside of the cell membrane that allows certain molecules to pass directly through the cell without having to go through a gap and not really going through the cell membrane at all. It's going through that window or the fenestrae. And of course, some very small molecules can go through diffusion. They can move through the plasma membrane directly. Then don't forget about endocytosis and exocytosis. That's another way, sometimes referred to as transcytosis. It's moving particles through a membrane, but it's moving it through by packaging that substance or molecule or chemical into a little vesicle and kind of allowing that whole vesicle to diffuse through the cell and then exit out the other end of the cell. So that's transcytosis. That's moving molecules directly through a vesicle in or out of the cell. Now, table 18.3 shows you 3 different types of capillaries, and they're based on how much tissue exchange or capillary exchange can take place. So the first category is called a continuous capillary. Notice that you don't have any fenestrae here. So you're not going to get a lot of movement in and out of this type of capillary. You can get some. You can still have some small molecules moving in and out, especially lipid-soluble molecules, but you don't have a lot of tissue perfusion here. You don't have a lot of capillary exchange here. So these are Pretty tightly associated cells making up the capillary. This is what your book calls least leaky. They allow for some substance to go through, but not very many. The second category is a fenestrated capillary. This is where you have a lot of little pores or openings through the cell membrane. And so you would expect a lot of capillary exchange going on here. Places like the kidneys, where you have, we'll talk about the excretion process and the reabsorption process a little bit later on. Places like your glands and your endocrine system, and in your small intestines, where you have a lot of movement of a substance in and out of the bloodstream, in and out of the capillaries. And then the largest of the openings that you would find in a capillary are called sinusoidal, because they have large spaces or large sinuses. They still have the fenestry or larger pore type openings. And this is where you would see a lot of movement in and out of a capillary of larger particles. Even whole cells can move out. So for example, the spleen is listed here. The spleen actually helps you get rid of some of your older broken down red blood cells. And that's because they have these large sinuses in the capillary that allows for red blood cells to kind of move out, the older type of red blood cells, worn out red blood cells. Now, looking at regulating capillary exchange out through the tissues is accomplished by a couple of mechanisms we'll talk about. Here we see something called myogenic mechanism. Myo refers to muscle, and genic is kind of automated here. And so this is an automated type of a reaction dealing with some muscles. And so this helps to control how much tissue perfusion you have, how much blood flow is leaving the capillaries and actually going out into the tissues. And so part of that is being regulated by little small bands of smooth muscle. These are called sphincters that really work like a rubber band would. It kind of wraps around the capillaries and either constricts or relaxes or dilates and allows for blood to either perfuse out, come out into the capillary network and out into the tissues, or bypass the capillary network and just go straight from the arteriole over to the venule. And so this is basically a mechanism in place that tries to automatically control this. So it is a stretch reflex. So if you have lots of blood flow coming through this little system, you don't want to overload how much, you don't want to flood the tissues with extra fluid. And so too much fluid here would cause a stretch scenario, a stretch reaction inside of this vessel. It would stretch out a little bit, causing some stretch on that sphincter muscle. That would help tighten down the sphincter muscle, and that would decrease how much blood flow is going out. And so it would kind of look like part B over here. Those sphincter muscles would constrict, and so now you would have blood flow going through the middle of the channel rather than out into the tissues. Section 5 also deals with movement of fluids in and out of the capillaries, and we'll kind of take a look at here. We'll look at a couple of different pressures associated with fluids running through or flowing through the vessels, and how much fluid is going to be moving out into the tissue. So it still relates with tissue perfusion here. And so we talk about a couple of different pressures. The first one is called hydrostatic pressure, which is really just the fluid, just a pressure of the fluid on the inside of a tube or the inside of a container. And so in this case, the fluid we happen to be talking about is the blood. The water, or the water that's in the plasma, is actually pushing, creating a force, trying to push on the wall of the vessel. It's almost like taking a beaker. I like the little diagram here. You've got a beaker of water. That water that's inside of the beaker has a higher pressure associated with it than the water outside of the beaker. But if you can imagine poking some holes inside of that beaker, then that's going to cause water to go from an area of high pressure to an area of low pressure. Well, that's kind of what's going to happen here. If you have an area of a capillary that has a higher water pressure than what's on the outside of that capillary in the tissues, then you'll have a tendency for the fluid, in this case plasma, to move from an area of higher pressure to an area of lower pressure. That's hydrostatic pressure. Another pressure in action here to help determine which way fluid is going to move either in or out of a capillary is something called osmotic pressure. Osmotic pressure has to deal with osmosis and it has to deal with how much solute or dissolved ions you have in a particular location. So your definition for osmosis is listed here. This is the movement of water. It's the diffusion of water, and it's water going from an area, starting into a solution that has a lower solute concentration, going toward an area that has a higher solute concentration. Water always wants to move toward the area that has the higher dissolved ions or the higher salt concentration, whatever solute that you're looking at. And so in the diagram below, the little blue structures here, these are all your little water molecules, and the little dots represent some kind of solute. Maybe it's sodium ions or potassium ions or something dissolved in that solution. And you see that on one side in solution A has a higher solute concentration. There's more solute ions dissolved there. Solution B is less concentrated, and so water wants to move toward the area that has a higher solute concentration. So it's going from a low solute concentration to an area of a high solute concentration. And so that's created that creates a condition of osmotic pressure. The definition there, the force we must apply to a solution to prevent water from moving into it by osmosis. In other words, this amount of solute here is essentially pushing back against the wall of this vessel, and so what happens is the solution here with the higher concentration, the higher osmolarity, has higher osmotic pressure. And that allows for water molecules to actually move in to that side. So if we start putting some of these pressures together, we see figure 18-13, part C here. You've got an arteriole on one side. You've got the venule on the other side. Out here would be your tissue area. This is where perfusion is going to take place. Here's a connecting capillary. On the arterial end, you have a higher water pressure. That's the HP here, the water pressure. And they give you some numbers to kind of work with. They say, well, the water pressure on the arterial side is usually higher, and they say it's about 35 millimeters of mercury. 35 millimeters of mercury would be that's water pressure, hydrostatic pressure. On this side, you have about 25 millimeters of mercury pressure created by the osmotic pressure. That's based on how much dissolved ions you would have inside of the bloodstream. On the venule side, notice that your hydrostatic pressure is lower. It's about 15 on the venule side. That's because you've lost water, you've lost some fluid, you've gone through some capillary exchange and some capillary tissue perfusion along the way. So as water or blood flows through this capillary, some of the water is going to leak out. It's going to go into the tissues. So once you get to the venule end, you see that your hydrostatic pressure is actually lower. Well, notice too that your ions are staying the same. They're not moving in or out. They're staying in the plasma or in the blood. And so your osmotic pressure from either end doesn't really change. And so your osmotic pressure on the arterial side is still about 25. The osmotic pressure on the venule side is also about 25. Now, one other pressure we need to look at is something called the interstitial fluid osmotic pressure. That's the osmotic pressure associated with any ions on the interstitial side in the tissue area. And so outside the capillary would be your interstitial fluid, and you have some ions there, but not very many. And so your osmotic pressure outside the vessel is only about 3 millimeters at this case. And so this is called your interstitial fluid osmotic pressure, is about 3. Then what you can do is also refer to what's called your colloid osmotic pressure. The colloid osmotic pressure is if you take basically it's a difference between your osmotic pressure inside the capillary and the osmotic pressure outside the capillary. So look at the osmotic pressure inside the capillary. That's 25. Your osmotic pressure outside the capillary, in this case, in this example, is 3. And so you could do a rough calculation and find out what the colloid, the overall, this is kind of like a net colloid osmotic pressure, and that's going to be 25 minus 3, that's going to be 22 millimeters of mercury would be your net osmotic pressure. So you have a little bit of an osmotic pressure gradient here because you do have most of your osmotic pressure inside. That's because you got most of your ions on the inside. You've got some ions on the outside of the capillary, and so you have a lower osmotic pressure outside. So to get your net or your colloid osmotic pressure, you just make that subtraction. Then lastly, if you put all of those numbers together, you have what's called the capillary net filtration pressure. This determines whether or not fluid is going to go out of a capillary or is fluid going to go into a capillary. And so again, on the arterial side, you've got higher hydrostatic pressure. And so to calculate the net filtration, you have to take your hydrostatic pressure, then subtract from that your colloid osmotic pressure. In this case, we said it was 22, and that gives you a positive number here. That's a net filtration pressure of 13, and that means that fluid is going to be moving out from the capillary into the tissues. Eventually, that fluid will be picked back up by your lymphatic system to try to return that back into circulation. On the venule side of things, you actually have some fluid returning back to circulation here, back into this capillary, because your water pressure drops. So on the venule side, your hydrostatic pressure has now dropped to around 15, and your colloid, notice that your colloid osmotic pressure is still 22. In other words, my ion concentration hasn't changed. I have the same amount of ions here, same concentration, and so my net filtration pressure actually is a negative number. It's a -7. And so that represents an inward movement of fluid. And so at the venule end, some of the fluid that's out here in the tissues will actually enter back into the capillary to return back to the bloodstream.